Hello students, all net aspirants. So in earlier video I said it is my effort to make you feel comfortable to read in between the lines of the text as per the biotechnology. Now the biotechnology applications. Okay. So keep the textbook with you and refer the text how my delivery is going to be synchronized with the text. And finally you are going to feel easiness and you are going to be benefited because different frames of the text is going to feed in your brain. So you switch over to the topic, that is biotechnology applications. In very first page information, the production of the biologicals, that means the products from the plants as well as animals. Biopharmaceuticals, that is the medicines from the living organisms. So production of biological and biopharmaceuticals from genetically modified organism, either microbe, plant or animal whose genes are modified, what we call genetically modified organism. And biotechnology having applications, having the uses, therapeutics, that is the products used for therapy, diagnostics for quick diagnosis of the disease, PCR, ELISA, recombinant DNA technology, and bioremediation using modified microbes to reduce the pollution like it is oil spills, and food processing like it is the yogurt and the cheese, the foods, and waste treatment, that is the sewage treatment, and genetically modified crops. These are all the biotechnological applications. Next, the critical area of the research, the three things: best catalyst in the form of improved microbe or purified enzyme. If you take some microbes like Pseudomonas fetida, it got modified, use it in case of the bioremediation process. If you take streptokinase, it is a purified enzyme. So, creation of the best catalyst in the form of improved microbe or purified enzyme first. The second, in order to produce the product, the catalyst to act to produce the product, we have to create congenial, favorable conditions to act to produce the product. That is the best bioreactors, the chemical engineered process, that is bioprocess engineering, like that. Third, best downstream processing, that is extraction of the compound, the purification finally. So undoubtedly the biotechnological products, it brought great qualitative improvement in case of the human life. Maybe in case of the food, maybe in case of the drugs, maybe in case of the veterinary products, in case of the animals. Next. And there are three ways to increase agricultural production. Agrochemical agriculture, that is inorganic fertilizers, gram or stick, urea, like that. And organic agriculture by using compost, farmyard manure and blue-green algae, what do you call it, is the organic agriculture. And third, it is genetically modified crops. We altered the genes in case of the plants and used to for some other purpose, like it is that is abiotic stresses and biotic stresses, to tolerate such stresses, like this genetically modified crops. So three ways. The first, next, and green revolution, increasing crop yield, the crop production, the food production to many folds, that is a threefold, what do you call the green revolution. So father of green revolution in India, Swaminathan. Simply father of green revolution, Norman Borlaug. So green revolution, it is because of the many contributed a lot for the green revolution. Maybe land reforms, irrigation facilities, economic help, improved varieties, agrochemical, better management practices, all contributed for green revolution. And partly contributed is the improved varieties imported from the other countries. And partly because of the improved varieties, mainly because of what? The two reasons: agrochemicals, that is fertilizer as well as pesticide, and second, better management practices. So partly because of improved varieties, mainly because of agrochemicals, pesticide, fertilizers and better management practices. Next, and Using agrochemicals, the limitation, the demerit is using agrochemicals is costly to the farmers of developing countries or underdeveloped countries. So beside which it causes for it is the pollution. So with the light of this knowledge of the genetics and molecular genetics, can you advise the farmer alternative methods to decrease the reliance, the dependence on what is the chemical fertilizers, chemical pesticide, and to reduce the pollution? Yes. And we can switch over from green revolution to gene revolution, because species specific barrier it is overcome because of discovery of restriction endonucleases, tremendous technology, that is recombinant DNA technology. So we can transfer genes between two unrelated organisms. Even you can transfer the genes from the bacteria to the plant. That is not possible by the conventional method. We cannot perform cross between the bacteria and the plant. But we can transfer the genes from the bacteria to the plant in non-conventional method, that is genetic engineering. So the plants, microbes or animals whose genes are altered, what do you call genetic modification, and organisms, such novel organisms, genetically different organism, what do you call it is genetically modified organism. What are the advantages of genetic modification or GMO, genetically modified organism, GMC, genetically modified crops? Besides resistance, that is must, besides resistance, it also has some other advantages. Abiotic stresses, that is tolerate against the drought, the cold and other stresses, like it is basmati. And improved nutritional quality. If you take the golden rice for vitamin A, it prevents night blindness. For production of the golden rice transgenic, there is the golden rice, we opted the type E, a variety of the rice, into which the three genes which are introduced, the genes from daffodil plant and genes from Erwinia bacterium, which was introduced for carotene. And the carotene is responsible, that is the precursor for the vitamin A. So because the carotene is available in case of the leaf, but not available in case of the grains, we require in case of the grains. So vitamin A in case of the golden rice. Next, post-harvest losses are reduced. If you take the tomato, transgenic tomato, that is flavor saver, bruise resistant, that is delayed ripening tomato. So we can increase the shelf life of the tomato, keeping tomatoes for fresh for long time in case of the shells, what we call shelf life increasement, flavor saver. Next, increase efficiency in utilization of the minerals, not increase efficiency in absorption, increase efficiency in utilization. So automatically that early depletion of the nutrients in the soil, it is going to be reduced. And pest resistant plant like the BT cotton, BT brinjal, BT potato, BT tomato, BT rice, like that, pest resistant plant. So we need not have to depend upon the pesticide if you created what is pest resistant plant. So in that, the example, pest resistant plant, if you take Bacillus thuringiensis, capital B and small t, Bacillus thuringiensis, some strains of the Bacillus thuringiensis produce toxic insecticidal protein at particular phase of the growth. What is that particular phase of the growth? During spore formation, that is sporulation. So certain strains of the bacteria produce toxic crystal protein at particular phase of the growth, that is during spore formation, that is sporulation stage. And it is species specific. It acts on only particular group of varieties insect. LCD, Lepidopterans, Coleopterans, Dipterans. Lepidopterans, tobacco budworm or mealworm. Coleopterans, it is the beetles. Dipterans, flies and mosquitoes. It is species specific, that is narrow spectrum insecticidal application. As the toxic protein coded by certain genes, suppose if you clone the genes of the bacteria into the plant, if you transfer the genes into the plant, we need not have to depend upon what it is, the insecticides or the pesticide. the plant itself got protected from what it is the pest, bt cotton, bt brinjal, bt tomato, like this. And conventional method of using these Bacillus thuringiensis, the spores of the Bacillus thuringiensis are available in the market in sachets, that is the pockets. And these spores, when mixed with the water, sprayed on to the trees, whenever insect ingest it, there is a solubilization of the crystal proteins in alkaline pH of the gut, because the proteins which are produced in inactive state, the what we call protoxins. So protoxins become active in case of alkaline pH of the gut, solubilization of the crystal protein, activation of the protein, and attach it to mid-gut epithelial cells. Next, creation of the spores, swelling, lysis, finally death of insect. So spores mixed with water, sprayed onto the trees, whenever insect ingest it, solubilization of the crystal protein protoxins in alkaline pH triggering environment, the activated protein attached to midgut epithelial cell create pores, swelling, lysis, and death of the particular insect. It is a conventional process. And in non-conventional process, we can transfer the genes encoding for the toxic protein into the plant. For this, choice of the genes depend upon crop and target pest, corn plant and cotton plant. And the bold cry CRY, it denotes what it is the crystal protein. The cry in italics indicate the gene encoding for what it is the protein. So cry 1AB meant for corn borer, cry 2AB 1AC cotton bollworm. So bollworm and borer, which are the pest, target pest. The plant, it is the corn and the cotton. The gene, it is cry 1AB, cry 2AB, cry 1AC. Okay. So in the choice of the genes depends upon crop and target pest. It is the pest resistant. And another pest resistant, that is nematode, which is parasitic on plants, animals as well as humans. And a nematode, the roundworm Meloidogyne incognitia, that is a parasitic on tobacco as well as tomato. It causes root knots and reducing the yield. Next, in all eukaryotes, there is a strategy, there is a mechanism against these pathogens, that is RNAi, RNA interference, RNA silencing, gene silencing. So it is a nucleic acid immunity inside the organism as a defense system. So particular RNA prevented from being translated. So there is no particular protein available for this nematode. So nematode cannot survive. So what is the source of the complementary RNA? The source of the complementary RNA, it could be from infection by virus having RNA genome, that is one. And second one is mobile genetic element, what we call transposons. Suppose the segment of the DNA it cut and inserted at other regions of the DNA, what we call transposons, jumping genes. And these transposons replicate via RNA intermediate. The first it is RNA is formed. By transcription RNA is formed. On RNA cDNA is formed by reverse transcription. As the cDNA again become double-stranded DNA and integrate into the host genome. So it is copy and paste mechanism. RNA is formed and cDNA is formed, double-stranded DNA formed and integrate into the host genome. As transposons only cut and paste mechanism, here it is transposons replicate via RNA intermediate, that is the copy and paste mechanism. So nematode specific genes are introduced into the host in such a way that it should produce sense RNA as well as antisense RNA. So what is the vector we are going to employ? Agrobacterium thumifaciens, that is a natural genetic engineer. It is employed to transfer nematode specific genes into the host in such a manner that it should produce sense RNA and antisense RNA. So we should introduce sense gene as well as antisense gene. Sense gene is going to produce RNA in which direction? 5'3" direction only. Antisense gene also produces RNA in 5'3" direction only. So two RNAs are formed, each in case of 5'3" direction. But in reverse case, the 5'3" and 3'5" is going to be complementary. So as it becomes complementary, double-stranded RNA molecule is formed. It prevents what? Translation. So which RNA is employed in case of RNA silencing? That is double-stranded RNA. It is employed in case of RNA, that is particular gene silencing. If you observe the two diagrams in case of the test, first and second diagram. The first one is typical control plant. Naturally, that is RNA interference present. It is also protected. And the right side diagram, even after deliberate infection, 5 days after infection, it got well protected. First diagram, it is having RNA interference. In second diagram, also having RNA interference. In first diagram, it is a typical control plant. Somewhat it is having the knots. And second diagram, it is also having knots, but it is well protected than what it is the first. Because even after deliberate infection, 5 days after infection, it remains, it is a normal, almost it is a normal, that is. So, the nematode can infect, but it do not infest, because there is a expression of what it is RNAi, that is RNA interference, gene silencing. So, it is a novel mechanism against what it is the pathogen in case of eukaryotes. And in case of ethical issues, as it comes in the botany part, ethical issues. Because genetic manipulation on one side it is having the positive effect, and other side of the coin it is having certain negative impact. If you observe in case of the paragraph wise, the ethical issues in case of the biotechnology applications, manipulation of any organism, whether plant or animal or microbe, should not go further without any regulation. There must be some regulation. So the regulation is not going to be restrictions. Some ethical standards are required to evaluate the morality of human activities. So ethical standards are required to evaluate the morality of the human activities. Because we are going beyond the morals. Because biological significance is also important. Their survival is also important. If after genetic modification, if you introduce into the environment, that is ecosystem, how it is going to have impact? There is going to be unpredictable changes in near future. So it must be some regulated. So regulation by the government set up an organization, Genetic Engineering Approval Committee. Genetic Engineering Approval Committee. It validates the GM research. To what extent the genuinity it is having the GM research. Whatever the GM products before releasing for the public, so get certificate from what the Genetic Engineering Approval Committee. So validate the GM research and products released for the public use, get the certificate from Genetic Engineering Approval Committee. That is one point. And in case of the next paragraph information, manipulation of organism for public use, the products like it is medical products or food products, created the problem with the patents granted for the same. Whatever the patents granted, it created some problem. Because there is a growing public anger towards many multinational companies, because the permissions are granted, patents are granted for those multinational companies for products and process. That products and process can make use of our genetic material, living organisms, which identified and used by the indigenous people or farmers of the particular area. So there is a growing anger towards multinational companies with regard to the patents granted, because for products and process they are making use of genetic material and organisms which identified by the indigenous people, the local people of that particular area. That is one thing. In next paragraph, if you take the rice, in Asiatic countries, the staple food, it is the rice. and richest diversity in case of the India itself, 2 lakh varieties of the rice are present. Next, a basmati, a variety of flavored rice for aroma and flavor. And this basmati is an Indian variety. How can you attribute that it is an Indian variety? It has been practiced, cultivated for centuries. That is one thing. Mentioned in folklore, ancient scriptures, poetry, it is our variety. But unfortunately, the basmati variety patent rights gone to other country, that is US. In 1997, a US based company got the patent rights for the basmati because we are unable to communicate the same as it is our variety, and it has been there for centuries, practiced for centuries, because all our scriptures and information present in case of Sanskrit language, as we are unable to communicate with them properly, and basmati, the patents gone to the other country, and it got extended to functional equivalence. That is, we are not allowed to sell basmati anywhere in the world, but US allowed to sell basmati everywhere in the world because they got functional equivalence, because they crossed our former variety basmati with semi-dwarf rice, and they got a new variety. They called it as a novel variety, some other names as the basmati, it is the texmati maybe. Okay, so it got to functional equivalence, and they also tried to patent the products and the process based on traditional herbal medicines like neem as a director, and also turmeric, curcuma. So if you are not vigilant, if you counter their application, reject their application by showing certain proper evidence, we are going to lose our legacy. And next, the biopiracy and biopatent. Whatever the bioresources, so taking bioresources by developed countries or multinational companies from developing countries or underdeveloped countries without any authentication, without any proper compensatory payment, what we call biopiracy. Because developed countries rich financially, poor in biodiversity and traditional knowledge. But underdeveloped countries and developing countries rich in biodiversity and traditional knowledge, but poor in financially. So our traditional knowledge with regard to the bioresources is exploited by the developed countries by the technical application skills. We save their time and energies in commercialization of this product. So developing countries now they are realizing that injustice made to them, inequalities, that is compensatory payment. So they realize that they are also making laws to prevent some such acts. Even Indian government, they passed on Second Amendment Act, keeping in the view of these things. The patent terms like emergency provisions, development, and research initiative, all these things. I hope just you all feel comfortable in learning all these things of the biotechnology and biotechnological applications. And you hear this first, listen the topic and practice it once, twice. You are going to be familiarized with these contents and certainly is going to benefit. Thank you.